All right, ladies and gentlemen, so welcome back. We are going to talk about DNA replication today. So when we talk about replicating something, we're talking about making a copy of it. So the whole point to DNA replication is to make lots of copies of DNA so that when your cells go through your cell cycle, right, we talked that G1, the S, G2 phase, before we go into mitosis and cytokinesis and make new cells, those new cells need a whole nother set of instructions in their DNA molecule so that they know what to do. So this is going to happen during our S phase of interphase. So we're going to kind of zoom in from where we were before into just this S phase of interphase and make a lot more copies of that DNA so that when we go to make new cells, all of those new cells that we create via mitosis or meiosis for that matter have a set of instructions to be able to follow so that they can function. So we're going to talk through the process of how DNA replication works, and we're going to keep it relatively simple. If we were to truly dive into this process, there are tons of enzymes, tons of molecules that are all going to make this process possible. But we're going to look at it from the more simplistic side of things. What do we really need to understand in terms of how the process works that are within the scope of what we're talking about for this school year? So I'm going to ask you to be responsible for two enzymes. All right, remember that an enzyme ends with that ending ASE. Okay, so that's how we're going to keep these guys straight. And the two enzymes that you're going to be responsible for knowing what they do in this process is helicase and this molecule called DNA polymerase. So let's talk about how this works. So remember that your DNA is a double helix. It's a twisted ladder. And you've got all of those base pairs, those A's, T's, C's, and G's that are hanging off here in the middle. And remember that they complementary base pair, right? So A goes with T, C goes with G. And so when we talk about DNA replication, the really cool thing is that that original molecule actually tells us exactly how to build the new one. All right, and how does it do that? Well, the first thing I need to be able to do is get access to the sequence of bases, get access to those A's, T's, C's, and G's, so I can know who to match them up with. So the first enzyme that comes in is DNA helicase. And helix, right? Like a double helix. The job of that DNA helicase is to unzip this DNA. So kind of think of it almost like a zipper, right? And if you ever looked at a zipper closely, you've got those little like teeth, all right, that kind of come together as you pull the zipper up or separate those teeth as you pull the zipper down. Well, DNA helicase is like pulling the zipper down, and it's going to pull open those teeth, or in this case, those bases, those A's, T's, C's, and G's that are hanging out here in the middle of that DNA molecule. And it creates this thing called a replication fork, kind of like a fork in a road, right? So that's where that shape is coming from. And those separate strands, once we open them up, can now give us a pattern to follow. So if I know I've got an A here, and a T here, and a C here, and a G here, I know what bases to bring in to finish the other side of that strand because they're complementary. So if I've got an A, I know I need a T. If I've got a C, I know I need a G, right? And so it tells me what to bring in next. So there's that DNA helicase, right? Opening up that replication fork and giving us access to the pattern to follow. The new bases then, all right, the pattern that we're following, those new bases got brought in by DNA polymerase. All right, so DNA polymerase, think about what that word means. Poly, right, we talked about polymers earlier this year. Monomers, like those nucleotides, are single pieces that we're going to link together to make a bigger polymer or a bigger molecule. So the purpose of DNA polymerase is to make a new DNA polymer. All right, and that replication is going to happen, that duplication is going to happen by following the bases that are on that original strand. So in this case, what we see here in red, and DNA polymerase is bringing in the complementary bases to build that blue strand. And it's going to happen on both of the DNA strands at the same time. So as DNA helicase is opening the strand, DNA polymerase is working on both sides of those two strands to match the pattern and create us a new DNA molecule. So by the time we're done with this, we take one DNA molecule and create two new DNA molecules from it. All right, and so there's that DNA polymerase working on the other strand as well. So DNA replication, if we look at the way that it works, we talk about it as being the semi-conservative process. Okay, well, what does that mean? Well, semi is kind of like half, and conservative, we're conserving or we're keeping part of it, all right? And so what this whole idea means is that I'm taking that original parent strand, okay, that original DNA molecule, and we're going to show half of it here in red and half of it here in blue. When that helicase comes in and that DNA polymerase comes in matching the pattern, that red strand from that original parent molecule here comes down here, all right, and then we create this new light blue strand as the new molecule. So here's one molecule of DNA that came from this. Half of it, that red strand, was from the original parent. That light blue strand is the new one that we just created. The same thing is happening on the other strand. So that darker blue is here. That's the original part of the molecule. And then that orange strand is the new one that DNA polymerase helped it to create. So what you end up creating is taking one DNA molecule and creating two DNA molecules from it. Half of each of those newly created molecules, half of the strands, come from the original parent. All right, so the red one coming from there, the dark blue coming from there. And then the other half, in case this one the light blue and this one the orange, are the new strands that we just created.
DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where. Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when. When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors. In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions and the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.